Good evening, everyone, and welcome to the last Research Tuesday event of 2023, XV Breathe Easier. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains, and the lands on which the University of Adelaide campuses here at North Terrace, at Waite and Roseworthy are all built. My name is Hayley Parkinson, and I'm undertaking my PhD in the School of Biomedicine here at the University of Adelaide, and I'm going to be your MC for tonight. Tonight, you'll hear about X-ray velocimetry, a game changer in lung health offering unparalleled imaging precision. Developed by our researchers, this breakthrough technology offers hope for early detection and tailored treatment, especially for conditions like cystic fibrosis. Tonight, we'll explore how X-ray velocimetry is transforming our approach to lung health and setting the stage for a brighter medical future. You'll hear from our researchers, Martin Donnelly and David Parsons. Martin is an associate professor in the Adelaide Medical School at the University of Adelaide. He's the leader of the Respiratory X-ray Imaging Laboratory and co-director of the Cystic Fibrosis Airway Research Group. You'll also hear from David, head of the Adelaide Cystic Fibrosis Gene Therapy Group at the Robertson Research Institute and an affiliate senior lecturer in the discipline of paediatrics at the University of Adelaide. He leads the cystic fibrosis stream at the Children's Research Centre and is Chief Medical Science in the Department of Respiratory and Sleep Medicine at the Women's and Children's Hospital. So without further ado, please join me in welcoming David and Martin up to the stage.
Thank you. Thanks very much. It struck me this morning that when talking about a new X-ray technology, I don't think there could be a better place to choose than the Bragg Lecture Theatre. And it's also hard to not have a little bit of imposter syndrome too, considering that Sir Lawrence Bragg received his Nobel Prize for work on X-ray crystallography at the age of 25. Anyway, today we're meeting by the beautiful Karawira Parry, which means Red Gum Forest River, and it's the Kaurna name for the River Torrens. So David and I also acknowledge and pay our respects to the Kaurna people, the traditional custodians whose ancestral lands we gather on today. So the Kaurna people are educators, and they pass their knowledge on from generation to generation. And I think there's a lot that we can learn from that intergenerational knowledge transfer. So my father, Brian, is a radiologist, so an expert in interpreting X-rays, and he's joining us online today. And he's passed that passion and that knowledge on to me. And I'm also passing this on to my kids who are here today, taking time out of their busy Snapchat and TikTok schedules to hopefully learn something new about what Dad does. So it's a real privilege for me to be able to be here to educate everyone about this fantastic new technology called X-ray velocimetry. So David and I have been part of the development of XV right from the start, and you're going to hear a fair bit of that story tonight. And as a result, we're pretty passionate about this technology, and we've purchased shares in the company 4D Medical, that are commercialising XV. And we're also going to use a few graphics from 4D Medical today because they clearly illustrate some of the points that we're going to talk about. So I thought I'd start off with telling you a little bit about who we are. So these are some of the Adelaide-based people that are involved in the work that we're doing. So on the left is the researchers from the Respiratory X-ray Imaging Lab and the Cystic Fibrosis Airway Research Group. And on the right are some of the WCH clinical team that look after the clinical care of all of our young kids with CF. So I'm probably biased, but I'm going to go out and say that the lung is the most amazing organ in the body. So did you know that on average, humans will breathe 11,000 litres of air every day? So that's enough volume to fill 200 kegs of beer. 200 beer kegs. And we also take about 670 million breaths in a lifetime. So we've got this thin sheet of muscle underneath the lung that separates the chest from the abdomen. And so when that muscle contracts, it moves downwards and increases the volume in the chest and causes us to breathe in. When that muscle relaxes, the opposite happens and we breathe out. And so the lungs keep us alive by moving almost constantly and mostly without us even noticing. So the role of the lung is to get oxygen out of the air and get it into the bloodstream. It's also responsible for removing carbon dioxide. So the lungs are this network of tubes, these conducting airways that start at the trachea and then they go out into the left and the right lung, and they split for 23 generations until they get down to these tiny little balloon-like structures called alveoli. And each of your lungs contains around 300 million alveoli. And in the alveoli, the gas is transported across from the air into the bloodstream, goes around the body where it can be used by all of your other organs. And then the carbon dioxide that is produced is also taken by the blood back to the alveoli, and then we breathe that out. So the lungs are able to achieve this remarkable gas transport while protecting us from all of the nasty stuff that's out in our environment, like bacteria, fungi, viruses, COVID-19 in some cases, and dust. And so all that stuff that we inhale with every single breath. So scientists now have a really good understanding of how the lung works, and right down to all these different types of cells that make up each component of the lung. So this is the surface of the conducting airways, and these are made up of epithelial cells. And in fact, you have variants of those epithelial cells in all of the tubes throughout the body, except for the blood vessels. They're slightly different. So the conducting airway epithelium is made up of more than half a dozen different cell types. And they're specialized cells that are responsible for regeneration of the airway, for salt balance, mucus secretion, and also for clearing mucus and anything that gets trapped in mucus, any of those things that we breathe in. And that mucus is part of the protective secretion for the lung. You all know what mucus is. It's that stuff that when you have a cold, your body's helpfully produce lots of it, and you have to either blow it out of your nose or cough it out of your lungs or constantly sniff it up like my kids do. So most people don't really think about how their lungs are working until something really actually goes wrong with them. So your body normally tells you when you're not able to breathe properly. So have a go. Try holding your breath for a minute. Hold it for as long as you can and notice what you feel. So what you begin to feel is that increasingly uncomfortable urge to breathe. And it's not really an urge that you can overcome. So why am I telling you this information? Well, it's really important for understanding the issues in the disease that we're particularly interested in. And it's called cystic fibrosis, or CF for short. So kids born with CF today now have a median life expectancy of 56. But many older people with CF aren't going to live that long and also have really real issues with quality of life. And most people with CF will eventually die from destruction of lung tissue. So think back to that feeling when you're holding your breath. Maybe if you are still holding your breath, you can let that out now. Don't want anyone to pass out here. So that feeling of wanting to breathe but not being able to is what people with cystic fibrosis have. And they have that every day, all the time, constantly worsening. So cystic fibrosis is caused by alterations in the DNA. So kind of like a spelling error in the DNA in a particular gene that's called CFTR. And cystic fibrosis is what we call a recessive genetic disorder. And it means that for a child to be born with CF, they need to inherit 2 faulty copies of the CF gene, one from each parent. So if your parents only have one faulty copy, then you won't have the disease. And you probably won't know that you're carrying one faulty copy. And also it's not infectious. So it means that if somebody with CF coughs on you, they can't pass that on to you. So this CFTR gene encodes for an ion channel in those epithelial cells that I just showed you before. And it's responsible for moving water across into and out of that epithelial surface. So CFTR maintains airway hydration by working with a second channel called ENaC. And I want you to remember ENaC because I'm going to come back to it a little bit later in the talk. So when the CFTR channel is defective, it results in dehydration of the airway surface. And what happens is that you get this production of thick, sticky mucus that sticks in airways and can block them. And it creates this perfect environment for bacteria to grow in. And you get this cycle of infection and inflammation that destroys the lung tissue and eventually results in lung failure. So you think that knowing all of this stuff about the lungs, that we would have really good tests to work out how well the lungs are working. But unfortunately, that's not the case. So the most commonly used lung function test is called spirometry. And it's quick, and it's all you need to do is blow into a tube, and it gives you a measure of how well your lungs are working. So amazingly, spirometry it's a technology that was invented back in the 1860s. So it turns out you don't need an app for everything, maybe. But spirometry gives you information about how much air your lungs can hold, as well as how fast you can get that air out of your lungs. So, but it does require a real effort. So you need to be able to blow as hard and as fast as you can to get all that air out. And so the test needs to be done in exactly the right way. And the issue is that means that it can't be done by young kids, so kids under maybe the age of 5 or 6. And that's really the opposite of what we want in cystic fibrosis. We want to know exactly what's going on in the lungs of our CF toddlers. And the reason for that is because we want to be able to prevent the decline in lung health, because prevention is always going to be better than trying to treat a disease, especially one that's as destructive as CF. So this is an example of the type of report that's produced by spirometry. It contains a lot of numbers. And one way that clinicians measure what's going on is by looking at what those numbers expressed as a percentage of what someone your age, weight, and height might have. So in CF, clinicians primarily use this measure called the forced expiratory volume in 1 second, or FEV1. And it gives an indicator of the level of obstruction in the lung. So if you think back about I showed that picture of mucus blocking airways. When that occurs, you can't get as much gas out of your airways. But spirometry doesn't tell you anything about where air is actually flowing within the lung. It's what we call a global measure of lung function, about how well your whole lung is working. And that's a bit of an issue in CF. Because it can be this patchy disease that can occur in different parts of the lung. And so we can't identify where any of those mucus blockages are occurring. And nevertheless, FEV1 is really widely reported. And the image on the right shows the median FEV1 of 3 groups of people with CF. So 10-year-olds, 18-year-olds, and 30-year-olds. And how that FEV1 has changed between 2002 and 2022. So you can see that for a lot of those people, lung function is improving, and that's the result of improved treatments for CF. And the great thing is that for 10-year-olds now, their lung function remains relatively normal, around that 100% mark. So what if, instead of measuring how the lungs function, we measure what the lung structure looks like? Well, chest X-ray is one way that we can do this. And to give you an example here, this is a front-on image and a side-on image of the chest X-rays of a 12-year-old female with advanced lung disease. So we've got a chronic lung infection with a bacteria called Pseudomonas. And the spirometry showed that they have an FEV1 of 47%. So that's half of what it should be. So she's really quite sick, and you can see there's like a cloudiness across the image that's produced by mucus plugging. You can also see there's that round thing there is an infusion port and a catheter for drug delivery. But the big issue with these two-dimensional images is what you're seeing is everything in that field. So you're looking at the lungs, the spine, the ribs, the heart, all overlaid over the top of one another. So that means that it's not sensitive enough to detect early structural changes that we have associated with CF. So currently what we call the gold standard is high resolution computed tomography, or CT. And that's used for identifying structural changes in the lung. And CT works by taking lots of images from around the outside of the lung in a special scanner. And then that's all fed into a computer algorithm that can work out what a slice through the lung would look like. And so, weird thing, although the lung functions by moving, when a CT scan is performed, the patient's told to hold their breath and stay as still as possible. So the nice thing about CT is that it produces a volume of data about the lung, and we can look at that data interactively in some different ways. And one of the ways is by looking at just these axial, coronal, or sagittal slices. You can re-slice that in other ways if you want. But yeah, looking from the top, the front, or the side, these common views. And the reason that I'm telling you this now is because this will be important for later on in the talk, because X-ray velocimetry that data can be viewed in exactly the same way. So these are examples of CT slices from two patients with CF before and after starting treatment with this relatively new-ish drug called Orkambi. So Orkambi is what we call a highly effective CFTR modulator. It works directly on that CFTR channel to help it function better. And these CT scans allow clinicians to see changes in mucus plugging in the airways. So in the bottom images, the arrows point to airways that are no longer blocked with mucus compared to those images at the top. And so what these structural changes indicate is that it's likely that these patients have improved lung function. But the big issue with CT is it has a high ionizing radiation dose. And that means that we can't do lots of repeat scans in our kids. So one of the tools that radiologists might use to be able to see an organ better is what's called a contrast agent. And these can be injected into the blood to help you see blood vessels better, or they could be inhaled into the lung to help see that lung tissue better. And so this leads to sort of a new area of research called functional lung imaging. And that aims to work out how the lung is working rather than what its structure looks like. And one method that's being used is this approach called hyperpolarized gas MRI. And so these are the types of images produced by that. They're xenon ventilation scans of a healthy 6-year-old at the top, and their FEV1 is 95% predicted, so maybe just a touch below what it should be. And then the bottom images show an 11-year-old with CF and an FEV1 of 102%, so a little bit above what you would normally predict. So even though that CF patient's lung function is normal, the xenon scans show that they have clear ventilation defects. There's patches of the lung where that gas can't get into. So while this approach is really nice because it allows us to actually see where those ventilation defects are, hyperpolarized gas MRI is really limited because it requires a lot of specialized equipment and it's relatively expensive to do. And so that means it's not really being used at all in CF and not in the pediatric space. So what this means is that there's a real need for a better functional lung imaging approach. So just to summarize, what are the gaps in measuring lung disease? Well, I'll give you a bit of an analogy here. So imagine that your car had a bit of a problem with its engine, and so you took it along to the mechanic. And the mechanic said to you, the only way that we can work out what's wrong is by measuring how much air is coming out of the exhaust pipe. But we can't do that in your car because we can only do it in cars that are older than 5 years of age. But they could also use some x-rays to show you what every part of the engine looked like. But they could only do that while the engine was turned off. So you'd think that was not a very good way of working out what's going on inside a really complex bit of machinery. And really up until now, the same has been true of diagnosing problems in the lungs of children. So spirometry doesn't tell you where a problem might be occurring, and it's not usable in that really young group of children. So we miss the earliest changes in disease. And CT provides images of lung structure while the lung is stationary. And that's really important because as I've noted, the job of the lung is to be almost constantly moving. And CT is also limited by radiation dose. So there has to be a better way of doing this, and we think there is. And I'm going to hand over to David now to talk about the beginning of the XV story.
Great, thank you, Martin. And welcome everyone tonight. Good to see you all here out this evening, and also to everybody online, which I think is up there. Well, I think Martin's very nicely pointed out the need for a way to understand how the lung moves and where the air is going, to really understand, in our case, if we have a gene therapy that works for CF, how do we know it's actually worked? Because the standard methods can clearly show things are normal when maybe they're not. And then you don't know where to go and treat, and you can't do it early enough. So let's see how we go here. So the beginning of this process started in Japan. Around about 2006, we started doing a number of different studies there, trying to work out different ways to look at the lung using X-ray light from the synchrotron here. This is the world's largest synchrotron. It's called the Spring 8 synchrotron. It's in the mountains south of Osaka in Japan. And we had, as I say, had a couple of different streams of research, but by about 2007 or 8, we started looking at how can we understand how the lung is moving? Because if we understand how the lung is moving in and out as you breathe, that's the same as the air moving in and out. And X-ray can show you things inside the body that you couldn't see otherwise. So what do you see when you look at, in this case, a baby rabbit? And on the top here, it shows a standard type of chest X-ray. It's called absorption X-ray. So what that means is as the X-rays go through the body, heavy things like bones show up darker and you can see the ribs and everything, and light things like air don't show anything and you can see really not much. On the left-hand side there, there's air in there, but you couldn't really do anything with that information. And if you go and look at some detail at the bottom of the diaphragm where you can just see a little bit of lung, well, there's bone, and the rest of it is like, yep, what do we do with that? So we need contrast. We need better resolution of what's happening in the air spaces to even think about this. And fortunately, there's a way to do that by changing the way you use X-rays, and it's done by a method called phase contrast. It's also used in light microscopy and so on, but here it's with X-ray. And the X-rays here at the synchrotron are so bright and so focused that we can look in the tiniest airways. And as you can see in a very young rabbit, look down to very high magnifications. But the difference here, bottom left, is you can now see the airways. You can also now see this sort of speckly pattern, which is what you can see also on the bottom right in high magnification. What we're seeing is a beautiful piece of bone there in the middle at the right, but we have a sort of speckle pattern is what we referred to it over the years. And that speckle pattern is what Martin said before. X-rays go through everything. So you're going through a small piece of lung and it's filled with alveoli. The X-ray goes through there and we see the integration of all those little alveoli as a speckle. We don't see individual ones, they're all on top of each other. So speckle's important, so keep that in mind. So with that, we thought, well, let's see what we can do. How did we get started? Well, this is a bit of history, you can tell from the hair colour. But this is after one of the studies in 2008 at the synchrotron. And fortunately, it shows all of the team, the major team that stayed together through the whole period. Myself and Martin on the edges, I guess appropriately in the middle, Andreas Sporus. So Andreas was a Monash engineer, aeronautical engineer that we started collaborating with around about that time. And he was interested in blood flow. He was at the synchrotron doing other things at the same time. And he was using a system called particle image velocimetry, PIV, and I'll explain that in a sec. But Andreas really was the lead of this whole project right from the start and right through to the end, which we'll come to later, or actually the start of the beginning of the end. But anyway, Andreas said, well, and we bumped into him in the hall, as sometimes these things happen, they're quite serendipitous. He said, well, I'm looking at this, and we talked about his problems and what was working, and we said, well, we've got this issue. We're trying to follow some motion in airways and, alveoli. And, okay. We swapped ideas, and we thought, well, this is worth following together. And PIV, that same method, was what he started with. He was using that in looking at blood flow, I should say. So what's PIV? Well, this is PIV. If you want to know how your car slips through the air at high speed, you can use a wind tunnel and special fancy light and particles of smoke and so on to produce things like this, where I think you can see they're all little arrows there moving as the air goes over the car, and it's a vector. So each little spot is a vector, and that means it's got a direction, the arrow, and it's got a colour, which tells you how big it is, how speedy is the air there. It's not just for cars, it's also for wings, whether it's old style fabric or modern metal. That's smoke running over there, and you can track those things using PIV. On the right here, biological. You can also track, in this case, the movement of a fish fin, and you can look at the turbulence in the water. So he basically said, well, let's regard each one of those little alveoli in that speckle pattern as a little vector that moves, and let's see what we can find out. So Martin's already mentioned beer once in the talk, so I can bring it up again. What's the first thing you do when you're trying to look for little tiny bubbly things? Well, you go to Guinness or Coca-Cola and say, can we even see these bubbles, and can we use that? So that's what we did, and you can see a beautiful Guinness glass replica there that we filled up with Coca-Cola, and the X-ray beam comes out of the back wall there, comes through this way onto the detector, which you can't see. So we thought this would work beautifully. It didn't, but it gave us enough information to say, okay, this is worth going further. So at this point, it's worth thinking about how do we actually do this? So if you can regard the lung as a set of Lego blocks, make it up like that. This is about 300 Lego blocks for that little picture. But now imagine that lung split into around about 6,000 tiny Lego blocks. And that's part of what the XV analysis method does. And you saw the vector maps that with the colour and the arrows. Well, if we took a slice through there using PIV as that lung moved in and out, then we can imagine in that speckle pattern we have a whole lot of little alveoli, and you're breathing in and out quietly there, and let the breath out, they're small. And then as you breathe in, they go bigger, and then they go back down again. So you should be able to track that motion as those little bubbles get bigger and then get smaller. And that's what you can get. This is a single slice, like the slice from top to bottom here, but just shows you an example of what you can get at. Now remember, and remember, for all of these images, they're 2D images, but things are happening in all directions, 3 dimensions. We can easily represent them this way. So with that, what do we get? Well, we take every one of those slices that the computer works out for us, combine them together to get, through the breath cycle, 4-dimensional lung X-ray velocimetry, XV. That's where it came from. And you might imagine why 4D medical is called 4D, because it's 4-dimensional. And some people sort of go, what's 4-dimensional? It's 3-dimensional, a volume, as it moves in time. So this is from 4D medical, and it's just to give you an idea. On the right here is a relatively healthy lung showing the changes in the colour patterns from red when you have no breath in your lungs, the bottom of your breath, and then you start to breathe in and it changes and goes larger. And it's not running as, yes, it is, there she goes. So no breath, breath comes up. And you'll notice on the right, it's mostly green, a bit of blue and red, but on the left lung has more blue, there's more ventilation coming in there, and on the right lung, there's not that much. There's something not right there. It's different, it's not sort of even and smooth. So it's not just about patterns and colours, all those come from the data. And the very first part of the data is some general numbers to give us an idea of what's happening using XV. So on the right here are just 3 examples of different types of information you can get as summary information. And we'll go through them. So when you think about each one of those Lego blocks starting off small and then getting bigger, if you compare the amount of space in the bigger area to what you started with, that is the specific ventilation for that small Lego block, and it changes with each position in the breath. It gets bigger and it gets smaller. And then there's 6,000 of them around the lung. So if you take all of those and put them together and say, how big were all of those? You get the top graph on the right. It's called a histogram of all of the ventilation in the lung, all the tiny areas of ventilation. You'll notice it's mostly in the middle. It's a bell-shaped curve. It's sort of common in biology. Most of everything about us, our height, our weight, whatever, has most people about in the middle, and there's outliers, very small, very large. And you can see it goes from 0 to 2, and in the middle is the centre. So that's where we get a mean specific ventilation. So it sounds fancy, but it just means what's the middle value? What's the average? And so it's sounding a little bit like the single values you get from normal lung function tests, a number that is supposed to mean this is how your lung is functioning. It's not, but it's just a starting point. The ventilation defect percentage, the pink blocked areas, is what that is. And this comes from other areas of imaging medicine, and it's the area that's less than 60% of the average ventilation point. So you can see it's on the bottom end of the graph, and the top shows, if you like, a normal sort of amount of low ventilation. The bottom one shows 15% as compared to 2.5, a lot of low ventilation over to the left there, and you'll notice the curve has flattened right out. So a simple way to look at that flattening of the curve is to say that's the width of the graph, and that's a measurement called ventilation heterogeneity. That's heterogeneity is just a fancy word for patchiness. And it's important because cystic fibrosis is a patchy lung disease. Children born with CF start with essentially beautiful, perfect lungs, and then something starts somewhere in that time between 5 or 6, maybe later, maybe earlier. And that's what we're trying to track. So, preclinical XV studies, this is where Martin's going to take over from me again. And it's often said you shouldn't work with animals or children. Martin's going to take the animal section, and I'm going to give you a little bit of information about the children later. Thank you.
Thanks, David.
So animal models are really vital for all types of biomedical research. Animals have fairly similar anatomy and physiology to humans. So mice actually share. Sorry, David's clicking, I think. We actually share more than 98% of our DNA with those animals. Animals are susceptible to many of the same health problems that we have, including cancer and diabetes and heart disease. And animals can also be genetically engineered so that they have similar diseases to humans. So, for example, we have cystic fibrosis rats here in Adelaide. And with a shorter lifespan than humans, we can actually study them throughout their life and through multiple generations. And it's really critical to be able to understand how diseases progress. So we've used a range of animal models here with XV, and you can see that despite their different sizes and lung shapes, XV can be applied to animals from mice all the way through to sheep. So we're really lucky here in Adelaide to have available to us this pretty fancy new machine called a Prometheum, and it's a small animal preclinical XV scanner, and we can use that for performing XV scans on mice or rats or ferrets. So it's a little bit like the synchrotron setups that we had in Japan 10 years ago, but it's small enough that it fits into this small enclosure that can go inside a laboratory. And we've used that to look at lung function in a range of animals. So what's inside that box? Well, this is a bit of a schematic of what's in there. There's an X-ray source at one end, and then a flat panel detector at the far end opposite that. And then we put an anaesthetized rat or mouse in the middle, and that's connected to a ventilator. And that makes it breathe regularly so that all its breaths are nice and even. And then the animal's rotated during that scan, and we capture images at a high frame rate at multiple points throughout the breath. And on the right, you can see us doing a recent study with the Prometheum, and that white cylinder in the middle is a tube that holds the anaesthetized and ventilated rat. And it takes about 5 minutes to do a scan. So I thought I'd tell you about one study that we've done recently in which we looked at beta-ENaC mice. And this is a model that develops cystic fibrosis-like lung disease. And the reason that we're using these is it's a little bit complicated. So we do have, as I said, we do have cystic fibrosis rat models available to us. But the weird thing about them is that although they have exactly the same mutation in their DNA that humans with CF have, their lungs are pretty much normal. So a little while ago, right back at the start, I asked you to remember ENaC. That's the epithelial sodium channel. So in the beta-ENaC mouse, that ENaC channel is upregulated. And what that means is that rather than the chloride channel not working, instead, if we make the sodium channel work a lot more than normal, then we produce a model that has the same dehydration of the airway surfaces and mucus plugging that we see in people with CF. So, I apologise, I'm going to present some graphs, but not too many. So what we did in this study was we first measured global lung function with a test that's kind of like spirometry that we do in our CF kids, but it's adapted to lab animals. And one of these measurements is called a pressure volume loop, and that measures how much the lungs can expand for a given volume that you put into them. And so in the beta-ENaC mice, so that's the green group there, you can see there was this upward shift in the measurements compared to their litter mates that are in blue. And so what that means is that their lungs are more compliant. So if you were to take a balloon and blow into it, imagine if you had two balloons, one of them was like a really soft rubber and it was really easy to blow it up. And the other one is much harder, you blow into it and it takes a lot of effort to blow. These beta-ENaC mice are like that easier balloon to blow up. So after that, we did some XV scans on them, and we found that all of those parameters in the XV analysis were altered. So all of those things that David has just spoken about. I'm only showing VDP here, but you can see that's increased, and that was that measure of how much of that graph fits into that pink box. And so that means that there's more areas in those beta-ENaC lungs that have reduced airflow. And importantly, these two parameters are correlated, so that meaning those XV results are related to what we're seeing in the lung function measurements. So the great thing about this is that we know we've already got altered global lung function from the spirometry, but here we can actually see where in the lung those changes are coming from. So these are, on the left are these axial slices, so sliced through the lung in that direction, and on the right are some coronal slices where the lung is sliced in that direction. And so there's roughly 30 of each there, and so we conclude that there's lots of information. So what I'm going to do is I'm going to break this down and make it a little bit simpler. So the control mouse has relatively homogeneous ventilation. So on here, on this map, green means average ventilation, blue means above average ventilation, and red means below average ventilation. In contrast, this is what we see in the beta-ENaC mice. So there's some airflow defects around the back of the lung, and then what we're seeing is some compensation around the front of the lung where we're getting above average airflow. So what this is showing us is that XV is sensitive enough to pick up these airflow defects and provides us with more information than what we would get from spirometry. So what we're now doing with this is that we're starting to apply this in a range of lung diseases beyond CF to see how they manifest. We're also starting to look at things and seeing things over time and seeing how diseases change and also how they would change with treatment. And being able to visualize this lung function in a totally new way is opening up lots of studies that we never thought were possible in the past. I'll finish up this preclinical application section with this important bit of news. So as part of a large medical research future fund grant that we share with 4D Medical and SAMRI and Uni Adelaide, we've had this large animal XVD scanner recently commissioned in Adelaide. And XVD means X-ray velocimetry device. So it's a dedicated scanner that's designed to acquire the images used for performing XV. Oh, so sorry, not a very flattering photo of our sheep there. But this was the first sheep that we've scanned with this machine, and so it was a really exciting day for us to actually get some data out of it. So this machine operates in a slightly different way to the clinical protocol that David's going to be talking about in a minute. It consists of 4 X-ray sources and 4 X-ray detectors. They can look at the lung from different directions simultaneously. And so that means that we can collect all the data that we need for an XV scan within one single breath. And so we have a whole lot of projects coming up that are designed to use XVD to learn about lung pathology in a range of animal models, including asthma and bronchopulmonary dysplasia, and looking at how the lung responds to things like endobronchial valve placement, which is a treatment for emphysema. So I'm going to hand back to David to talk about some of the exciting findings from a feasibility trial that we've just started in our kids at the WCH.
Great, thank you. So our interest is in how can we make life better for children born with CF. So we have this technology that we can use to look at all sorts of things in animals. But how does it work in people? Well, it's already available out there, not on Medicare, but it's a test that your doctor could prescribe for you if you wished as an adult. But what's important to us is what happens, how can we apply it to children, and particularly, can we and how can we apply it to children soon after they're born? Because that's when, that's the period in those first few years when things are going to start to happen. So briefly, the goals of the study that we have underway at the Women's and Children's Hospital across the road is a feasibility study. It's a small study to just begin things. And the primary outcome is to determine whether we could do these types of imaging studies as they're done in adults. Can we do it in children? Children are not just small adults. They have their own special needs. Their lungs develop at different, they're developing still. There's a whole lot of factors that come into it. Secondary outcomes about how do can we tell with XV technology, how does it compare to our standard spirometry, those sorts of lung function techniques? Also with the structural disease, say the assessment that you could get from CT, well, we're not really looking for structure in what we do, but we do get structure because we know where in the lung things are happening. So some idea of a comparison to the direct structure and what we get out of alongside our functional imaging is going to be interesting. And also, obviously, to work out, is the approach using XV in children safe and is it effective? Is it appropriate? So that study is, as I said, quite small, 20 children who have CF and 20 children who don't have CF. The imaging takes about 10 to 15 minutes, of which the actual imaging part is about 5 minutes or less. They get the standard lung function tests on the same day, taking somewhat longer, and that's spirometry, which you've heard about, lung volumes, which is about how much air can you get into your lung, how much when you stop breathing, how much can you push out, all those sorts of things that tell you about the lung. And lung clearance index, which is a method that allows you to follow how gas exchange occurs over the lung, and you get a single number about how air is moving. So it's all global stuff. And in this study, they need a pre-existing CT scan. So the XV protocol here, as Martin mentioned, is different to what's used in animals. And the setup here is using a standard C-arm fluoroscope. You can see that top left. And that's an X-ray machine that will rotate. And it needs to take 5 different angles of the subject. And then that goes off to be processed by 4D Medical. The processing involves taking those images from the 5 angles, combining it with an understanding of where the lung is in the chest cavity and where the parts of the lobes are in the chest, running it through the same analytical routines that are used for the animal studies, and then getting all of those slices through the lung. So on the left here, and on the right, you will have seen the one on the right from Martin. This is trying to get an idea at this very early stage of what healthy looks like, because if we don't know what healthy looks like, we don't know what is abnormal. And that's part of what we're going to get out of this study, that sort of comparison. But on the left, you can see here's a 46-year-old adult male, and this is looked pretty reasonable as a healthy control indication. And you can see top left that there's a fair bit of green average ventilation, a bit more red, a bit more blue, but the histogram below it shows it's roughly that bell-shaped curve. Animal models are useful too as normals because they're mammals just like us. Their lungs function essentially the same in terms of evolution, and so we can get an idea, because we don't have any data yet for healthy children. And so looking again on the right, now if you look at that greenness, the blueness, the redness, you do get a sense of, okay, it's a bit different here and there, but you have a sort of feel for what it should look like as healthy. So let's go to one child, it's actually a teenager, 17-year-old male who has CF with the most common variant out in the population of those with CF. The lung function test is shown on the right. Don't need to look at any detail there. It's interesting to note that the FEV1 is 96%. That's normal. That's healthy. The spirometry, diffusing capacity, lung volumes, lung clearance index, everything here is normal. And clinically, this teenager was clinically well at the time, had previous hospitalisations, as happens in CF, that had recovered well, and various other medical issues that are not unusual for a teenager with CF. So, there we go. CT, as I said, there needed to be a pre-existing CT so that there's no additional radiation exposure, because CTs have a lot of exposure. And the radiology report, lung appearance is unremarkable. Nothing particularly going on there. And a previous fungal infection, and that's something that children with CF can get and can cause quite a large problem, has been resolved. So with all that information, you're thinking, okay, well, what will XV show us? Well, that's on the next one. And it struck me just while Martin was talking then, it's starting to look a bit like the beta-renac mouse, the one that had the lung disease. You can see here on the left, and we don't need to worry about the numbers because they're general average numbers of most things. But look at the graph there, the histogram. It's almost completely flat. We've lost a lot of average ventilation, and there's a lot more high-level ventilation, airflow, that's the blue, and a lot more low ventilation, that's the red, and look where they're located. That's the really cool thing here, is you know where it's happening now. It's not just something measured at the mouth as a single number. So on the right, from this same case study, you can see a few samples from the axial, this way, and the coronal slices. and they really just echo what you can see on the left. And so we have, sorry, I forgot to push buttons while I was going there, the two examples there of the high ventilation and the very low ventilation. The thing that has got me really excited, and I think has amazing capability for the future, is following the airflow during the breath. It's nice to have, the static stuff and the summary stuff, but now we can know during that breath how things change, whether it's going in or going out. I'm only going to talk about the inspiration here because of time, but you can see when this starts, think back to those Lego blocks. You start with no ventilation and then you expand. And what do you see? It starts with red, no ventilation, comes up in general, but notice it's not suddenly all blue in that very blue part. It takes a while to get there. So having the tools to understand what happens during a breath is going to be absolutely fantastic to see in CF as well as any other lung disease. And so what's the future of XV? Where are we heading from here? Where do we think it's going to go? Well, we think the future is bright, and that's an imaging pun that everybody uses, but I felt it was important to bring it up here because of the use of light, X-ray light. Back in August, we were fortunate as a team to win the Excellence in Interdisciplinary Scientific Research Award from the Eureka Prize 2003 people. So we obviously had a bit of fun afterwards. On the right here, I do need to go back to that picture way back in Japan. I forgot to mention Kay Morgan, who was sitting on the right. She's here. She's throughout that period been fabulous X-ray physicist, synchrotron driver, just an amazing colleague that's allowed us to actually do all this work. And in the left-hand side of Andreas, who I also forgot, Steve Dubsky, who was critical at the start to take all those Lego block bits of information and work out how to bring them together, to analyse them, create the software to do the job. So it's really a team of 5 that we're talking about here for this Eureka Prize. They couldn't, unfortunately, Andreas and Steve couldn't be there for the time. So what we see we have here is a wonderful translational research capability that will allow us to take information from small animals, large animals and humans and use the same technology all the way through to understand what's happening. We don't have to change because of size. We don't have to change because of species. We can put all that information together. And here at the bottom is the XVD in the human imaging setup. It's like the machine we got a few months ago, but it's designed for someone sitting up rather than a sheep lying down. And you'll be able to notice Andreas here has the same hair colour that I have now, so time has moved on. So what are our next steps? It's a key part of our MRFF grant is to move to developing imaging so that we can avoid CT, because then things start to open up. With the XVD machine, we're changing from a fluoroscope which does all sorts of things in a hospital to do with X-rays to one that's only focused, I guess that's another pun, on lungs and reducing X-ray dose and giving us exactly what we want in following motion. So all of these things on the right, some of them we've started work on, some of them haven't been touched yet, but we're going to get an amazing amount of information out of all these analyses. And so extracting deeper, detailed information is going to be crucial. As I mentioned, we've got some numbers, we've got some nice colour maps and graphs and that, but that's not really telling us how the lung's functioning. And one of the questions is, what new measures can we take out of that huge amount of data with every image that we get? And so we've already engaged with artificial intelligence researchers from the university and the hospital to start that process of looking deeper and understanding better. We want to also, as part of that, whatever comes through, we need to be able to present that data to busy doctors in a way that's better than a single number, that can sort of encapsulate the complexity of the lung and the different complexity when disease is present. and apply this XV to other lung diseases. We've talked about that. I like to think of it as, well, anywhere where airflow is important in lung disease. And if you think about it, well, that's it. That's all of them. So before I finish, I do want to bring up the acknowledgements for all of the, all the people in and around our team over the years, along the bottom line, the various funders that have put in support from, I guess, probably about 2008 or 9 when we first started. I particularly want to point out the Cystic Fibrosis Federation of Australia on the right at the bottom there. They provided the first real funding, good level funding for us to begin our studies at the synchrotron in 2010. So, cycling back to the start, this was about XV and breathing easier. The XV capabilities, I think we've been able to show you, help us know how the lung works during breathing, not just what it looks like. It shows us where the airflow is abnormal. It's got high sensitivity, and we don't need to use any special contrast agents or manoeuvres, ways of blowing, and it means once we've dealt with the radiation dose aspects for children, we can start going into younger ages. So our vision, or my vision, I think probably our vision for what could we imagine for the future here for children with CF? Well, my perfect vision is a child is born with CF. We know they've got pretty healthy lungs to start with. We can start looking at the lungs early to, I expect, find a normal healthy lung and do regular XV imaging until something pops up and be able to go and treat that because you know exactly where it is. And then track that disease progression, or actually, since we're treating it, track how well the treatment's going. Say this worked or it didn't work. Use a different drug, our gene therapy or a pharmaceutical, and ultimately get down to where we can prevent the lung disease in CF ever starting. So overall, we want to improve the course and the quality of life for children with CF. So thank you for your time, and Martin and I be pleased to answer questions.
Thanks so much, David and Martin. That was really fantastic. It was a really interesting talk, and it's great to see that we're getting these new fantastic technologies in Adelaide to really improve the lives of people with CF. So we'll now open the floor to questions. So I do have a pre-submitted question here, which I'll ask first while you guys get your thinking caps on. So the pre-submitted question is, would this technology also be more useful than CT scans for pulmonary fibrosis? And then the second part to this question is this technology already publicly available, or is it still in its sort of research phase?
Okay. The first part of the question is, would it be useful for pulmonary fibrosis? All I can say is probably, because the work hasn't been done. It's another disease where airflow will be important. So in a general sense, I think it will be useful, but I have no data yet to say anything about that. The second part was, is it available? Well, I mentioned earlier on, it's been commercialised by Andreas Fouras. He formed a startup company 2015-ish, Melbourne company, and he has commercialised it for use in adults. It's also approved in the US as well as much as in Australia for use by adults. So it's out there. It's not just a nice research idea. It's gone all the way over 12 years from a great idea from Andreas to something that's out there being used.
Fantastic. Do we have any questions in the audience? If you just raise your hand, we have some roving mics coming around for you to ask a question. There's just one coming down here.
One down below.
Good. Thank you for that presentation. The chloride, with these mutations, the chloride channel will be affected in every cell right throughout the lung. Is it predictable that only one area of the lung seems to be affected from an airflow viewpoint? You've only got that one little red area down the bottom. Was that predictable or understandable?
No, it's completely unpredictable. As we said, CF disease is patchy as it develops, and it's thought that that's probably probably comes from, okay, everything's okay to start with, but there's that defect in how the airway is hydrated. So somewhere in the lung, I'm imagining here, a little bit of mucus gets stuck or partially blocks the airway, but it can't get moved out because the systems don't work properly. And then that starts to build and bacteria land there and everything. So you have an area like that. But there could and probably are going to be other areas all happening the same or different times. That would be how CF develops. That's my thinking of it. How about it?
Yeah, and I think one of the things that David mentioned was looking at how that correlates with structural changes. And so one of the things that we're going to be doing in this pediatric feasibility study is comparing to a thing called PRAGMA CF. So it's I wouldn't say commonly used, but it's a method that some people use to be able to score different parts of those CT scans and be able to say, yep, in that area we're seeing a structural change that we can then hopefully correlate with what we're seeing in our XV.
I think someone down the front here had a question. If you just pop your hand up again, thank you.
How does knowing the area of the lung that's affected with CF, how does that actually change your treatment?
Not sure.
How does knowing where in the lung is affected help change your treatment?
Oh, okay, yes.
So it can allow clinicians to do more targeted treatments. It allows you to be able to keep track of what's going on in those areas. It allows you to do bronchoscopy to be able to.
And so bronchoscopy is, for those who don't know, it's a flexible tube that you can put down into the lung, look around, do things, treat, take little biopsies and so on. You can also deliver things and right now if you have, let's say, Pseudomonas infection in part of your lung, and we can find out where that probably is because that airflow will be different. My vision for the future is, well, we know it's there. We can now go and treat that bit of the lung with a very strong antibiotic without having to treat the whole person or the whole lung with that same strong antibiotic, which always has side effects. So I see that as a real extra advantage, that the ability to know where things are allows you to know better what to do and gives you more choices. Does that answer your question? Yep, okay. Oh, I see, yes. Yep.
Do we have any more questions? Pop your hand up. Oh, go on, just here.
Thank you. First question, somebody with bronchiectasis, would this technique of an adult, would this technique help treat an older person with bronchiectasis?
Well, it wouldn't help treat, it would help you understand better how much disease you have and where it's located for whatever treatments are available.
Can I just add, for those that don't know, bronchiectasis is common in cystic fibrosis. It's this irreversible dilation of airways. So the airways actually become larger in the lung. And with XV, we should be able to see that that's occurring and then track those changes over time. So as David says, not necessarily as a treatment, but allows you to know exactly what's going on in the lung.
Second question. Somebody with a small external expiratory volume, possibly asthma, would it help in any way to determine where the problem in breathing is?
It would help you know perhaps where things are focused, but it wouldn't directly be a reason to change treatment. That would be something for your physician to work out using that information as well as your lung function test, as well as CT and so on. So I think at the early phase of this development in going out into the medical sphere, it's going to be additional information, an adjunct to what's already being used. But I guess certainly for us in CF in children, we're looking for it to fairly rapidly, and what's rapid? Well, I don't know. But it takes a long time to change things in medicine. So I think in the next 10 years, we're going to see things starting to happen in children. That's really going to change the way we treat. So in that sort of sense, there's going to be a change in treatment because you're going to know what you're doing and you'll be able to see when it starts.
I can probably add one more bit of information to that we've got this great new technology, but at the moment, we're not completely sure what XV looks like. So what do those airflow maps look like? What do the numbers look like? How do they change? We don't know that yet for every disease. And that's one of the places where the animal models are really useful because they allow us to start to see in an animal what that's like. But there's some challenges with doing that in the human population at the moment. And that's why we've started the feasibility study in kids with CF so that we can start to say, this is what it looks like in this person with CF. This is what it looks like in a child that doesn't have CF. So I think we don't yet know what it would look like in that case with asthma. But I think David's point that any disease that changes airflow is going to change what we see with XV.
We have an online question here.
Thank you. This is actually a personal question. Could this be a tool down the track to help educate smokers and perhaps catch early on the effects of what they're doing to their body?
That's a good thought. I often wonder about the idea that education will change people. More recently, politically, it makes no difference at all, and people push away from it. So I would hope so. It used to be what I thought, well, more information, better, clearer education will help. Smoking is an extraordinarily addictive disease, and it really is a disease. It's hard. I think it'd be useful, but I don't know how much impact it will have. It's just part of the range of tools for smoking prevention or cessation.
I'll add a little bit more to that as well. Go back to what we said before, that the lung it's so resilient. Like you can do so much damage to the lung and it still manages to function and keep you alive. And so you can take smokers that have smoked for years and they'll still have an FEV1 that is close to 100%. So it would be fascinating to be able to take a tool like XV that we hope is more sensitive to be able to say, hey, your lung is not normal. This is what has actually happened from that smoking. As David says, though, whether that actually changes anything is a different question.
We just have another one in the middle here.
I can see a role of XV in CF as far as their CPT therapy. If you can identify if it's the top, middle, the front, the back, or the location, or the left or the right, they can actually change or modify their therapy, the number of repetitions, and they can actually see how effective long term that therapy, and maybe modify the therapy to improve their lung function.
Yeah, exactly. And in fact, one of the areas that we have on our slate of things to do is to collaborate with physiotherapists in CF to say, well, okay, first off, what changes when you do, general physiotherapy? Does it actually move things in or out? What's happening? And then your point is really good to say, well, we can see it's here. So don't worry about the rest, target everything in this position. Yeah.
Hello, David. I am sitting here now 20 years on from when I was working with you, and it's nice to see the mice are still there. We were putting viral vectors into mice and so forth. It made me reflect on the lunchtime meetings we had with the doctors in the department, and they weren't overly excited about us putting viruses into diseased lungs of CF patients. Are they more excited about what you're doing now?
Well, the environment has changed a lot from the time you were with us, as you know. With the advent of the modulator therapies, the drugs that are taken twice a day, and Martin mentioned one, Orkambi, there's a whole range of others, that has been an amazing change in the health of lungs for children and adults with CF by taking that drug. And what it does is work on how that channel works and how the channel moves water in and out and so on. So that's really changed the environment. So, let's say 90% of people with CF, roughly speaking, will have some benefit from one of these new drugs. Some of it's fantastic benefits, some of it's not so great, some are enough at all. So 10 or 15% of people with CF don't have a type of gene mutation that can be treated that way. It's because the easiest way to think about it is some people have no CFTR, so you can't make it work better. So we're still in the game because there's a whole group that can't be treated right now with those fantastic new drugs. If what we do works for that smaller group, then that will actually also work for all the rest of the group. What's going to be the future for CF drug treatment or gene therapy? I think things are going to be a mixture of everything. We're going to take the best of each of the technologies and put them together at different points over time to come out with a treatment that does the best for an individual, because it's the time of personalised medicine, working out which particular gene mutation you have, what are the other factors that are feeding into that expression of that gene, what sort of level of disease, can you still breathe properly? XV can tell you all the areas you can get to, things like that. So I think I ran off the question a bit there, but did I answer that? Are we still?
I think the clinicians are excited.
Yeah.
I think you only have to look at that and the report that David showed that said, yeah, this child lung function is normal, all of their measurements are normal, their CT is normal, but that XV report is not normal. You can really quickly see what's going on in that. So they're excited. Being able to see how things change over time in those patients is going to be the real game changer, I think, to be able to see, yep, in an exacerbation, this is what your lung is doing as you get better with a therapy. That's what's happening.
And the genetic therapies, just to jump back to the gene vectors and so on, dosing, they work, but they're very hard to get in. The lung has been designed through evolution to keep everything out. So as you know, we're trying ways to work out how to sneak past the barriers to get to the cells with the gene that we want to put in to correct things. That's got better, but it's still on a slow curve upwards, fortunately. So there's a whole range of other gene vectors from what we worked on that are now taking a big part of the direction of gene therapy for CF lung disease.
All right, I think we have another online question, and for the interest of time, we might make this the final question of tonight.
Question from one of our guests is, how long will it take for this to be a preferred diagnostic tool in the medical profession?
The wonderful how long will it take question for medical research. I have truly no idea. As I mentioned, things change slowly in medicine. There are certain ways that things are done, and it's like most areas of life, it's a bit hard to push in new ideas and new ways, but things are moving, I think, at a good pace for the adoption of XV. It's out there already for adults. It's showing the sorts of, I guess, if you like, the initial biases of what we might see, what we might not see. That's sort of coming to be, yeah, that actually is like that. The standard joke is 5 to 10 years. You say that for everything because we just don't know. It's going to depend on how XV is taken up in different places in the world. I mean, there is an enormous need for good diagnostic tests in respiratory medicine worldwide. It's a massive amount of cost in there at the moment across CT, MRI, PFTs, something else I can't remember. Sort of 4 major areas of testing the lung. I think it's going to move quite quickly as an adjunct, but I don't think it'll ever take over because there's too many other useful bits of information that are out there that inform the clinicians, well, what's going on and how do we best look after you?
Fantastic. Well, I think we might wrap it up there for tonight. So please join me once again in thanking our fantastic speakers.
Thank you.
And of course, thanks to all of you for coming tonight and those of you who are watching online as well. Be sure to sign up to our mailing list to get the latest info on our Research Tuesday events. And given it's the last Research Tuesday of this year, I just wanted to wish everyone a really fantastic holiday season. And I'm excited to see some of your faces next year to learn about the really exciting research we have going on here at the university. So thanks for coming.